the middle part of chapter 16. I've watched the eye anatomy lab and have gone through that because we're talking all about the eye and vision and visual pathways today. This vision is a little bit more complicated than smell and olfaction and gustation, so smell and taste. And that's why it kind of has the whole lecture for itself. So we're going to start with, we've already done some of these things like learning objectives. We talked about a lot of the structures of the eye in lab, but we're also going to talk a little bit about refraction, how we focus the light on the retina, and then phototransduction. So like how exactly those rods and cones in the eye are actually going to be taking the light signal and transducing it or changing it into an electrical signal that the brain can understand. So here is just kind of a review. This is what our eye looks like. If we look at light, it's going to be coming from outside and going in, right? So our eyes are special senses. They're found in the head. They're exteroreceptors, right? We're receiving things from outside the body. And then the cells at the back of the eye, our photoreceptors, are going to be photoreceptors, right? They're perceiving light, and then they're transducing it or changing it into an electrical signal or an action potential. And it's going to go back through the optic nerve to the brain. And that is, remember that the brain needs it to be transmitted from whatever modality it is, you know, if it's sound or chemical or light. We need to change it into an electrical signal so that the brain can understand it. So our light is going to come in and it's going to go through, I want you guys to know the order that light passes through structures in the eye. So we have talked about what these structures are called. It's going to go through the cornea first. So the cornea is clear. The cornea does not have blood vessels in it because it's clear. We don't want any blood vessels to like obscure our vision. It actually has some epithelial cells on the outer layer and on the inner layer, and then it's filled with some like non-cellular stroma on the middle. Inside we have the aqueous humor. The aqueous humor is going to like nourish the inside of the cornea, whereas the tears are going to nourish the outside of the cornea, those cells on there. So remember, the aqueous humor is going to be created here by the ciliary bodies, and so it's going to go out here into the posterior chamber of the anterior cavity, and then it's going to go through the pupil, and it's going to come here into the anterior chamber of the anterior cavity, and it's going to be reabsorbed at these scleral venous sinuses. So that's just a quick recap of that aqueous humor circulation. So this is like that watery fluid that fills the front of our eye. The iris here is going to be the colored part of your eye. So it gives you, if you have, brown eyes or blue eyes or whatnot, that's the iris. And it has a hole in the center called the pupil. And this pupil is going to be, the iris and the pupil are really important in regulating the amount of light that can enter the eye. And so we have the pupillary muscles. We talked about our sympathetic and parasympathetic innervation. So sympathetic innervation is going to cause them to dilate, and that's going to let more light in. Parasympathetic is going to cause them to constrict. It's going to let less light in. And it's also going to affect where the light kind of hits the lens. And so when it dilates, the light will kind of hit the whole lens, even these little outer areas, which are sort of thinner. Whereas if they're constricted, then the light is only going to go through the middle of the lens. And the middle of the lens is kind of the clearest part of the lens. So our light is going to come, it's going to hit through our cornea, and then go through the aqueous humor. It's going to go through the pupil, which is the hole in the iris. And then it's going to go through the lens. The lens is really important because the shape of the lens, whether it's round or whether it's flat, can actually determine how much light bends or the refraction of light. And that's going to, that light is then going to focus somewhere in the eye. And hopefully, if everything works out, then it should focus directly back here on the fovea centralis. So it has to go through this thick vitreous humor. It's a gel-like vitreous humor. And this is made during embryonic development. Unlike the aqueous humor, it's not continuously made and reabsorbed. This is just always there, your vitreous humor. So the light goes through here, now it hits the vitreous humor, and then it's going to hit the retina. And it's going to focus at this fovea centralis. And we said fovea means a little pit. Like we had the fovea on the top of the femur. And so this is where we have most of our cones. We're going to talk about the different types of photoreceptors, but our cones are going to be gathered, or most of our cones are going to be found at the fovea centralis. So whenever you're focusing on something and it's really bright and clear and in color, it probably means that the light is being directly focused onto the fovea centralis, where all of your cones are. And this is the fovea centralis is in the very middle of something called the macula, or the macula lutea, and that's going to be sort of this whole area around the fovea centralis, and that's going to have, as we said, the high proportion of cones in it for color vision. Excuse me. We talked about the pupil already, and how the pupil or the iris is going to control the pupil diameter. So the sphincter pupillae muscles, these are going to be parasympathetically innervated. They're going to constrict with parasympathetic activity from cranial nerve 3. Whereas the dilator pupil muscle are going to be the muscles that dilate and like they open up the pupil, and they are going to dilate with sympathetic nervous system activity. And then you do have a reflex where reflexively your eye will do this. The more light that hits the eye, the more your pupil will constrict. And that's going to like regulate the amount of light in your eye, so you don't have so much light hitting the back of your eye. If we have darkness, like if you're in a dark room, your pupils will dilate to let more light in so you can see better. So now I want to go and talk a little bit about the retina again and kind of refresh our memory of what we can see in the retina. Remember, the retina is the very back of the eye. This is where we're going to be focusing the light. And we have 3 different layers. We have the photoreceptor cell layer. Let's see, do I have a picture of that? Not here. We talked about this already, and there are the pictures of this in our anatomy lab, and we'll see that again. So the photoreceptor cell layer is the this is the layer where we actually have photoreceptors, where the phototransduction happens. It contains rods and cones. Then next we have the bipolar cell layer. Remember that bipolar neurons are the ones that have one axon and one dendrite, and they're kind of special. They receive input from the rods and cones and send it to the ganglion cell layer. And these are going to receive that input, and then the axons are going to go out and exit the eye through the optic nerve and send it up to the brain. These are the main 3 you'll need to know. There are some other interneurons called horizontal and amacrine cells, but we don't really, I'm not going to focus on them as much here. I mentioned the vitreous and aqueous humor. As a review, for the aqueous humor is found in the front of the eye, and it's watery, whereas the vitreous humor is the back of the eye, and it is thick and gel-like. So these are like the fluids that help the eye maintain its shape. The aqueous humor, the watery humor in the front of the eye here, is going to be always continuously created as well as continuously reabsorbed, just like your CSF. So the choroid plexus here is going to be creating it. It's going to then, it kind of filters your blood plasma through here. So now we have this aqueous humor that comes out in the posterior chamber of the anterior cavity. Then it's going to go through the pupil, and it's going to come up to the anterior chamber of the anterior cavity. And then it's going to, nourish the inside of the cornea as well as nourishing the lens. And then it's going to get reabsorbed at the scleral venous sinus. If we have problems with reabsorbing this, we're going to be making, if we make more than we reabsorb, then our eyeball can actually be like a water balloon and get more full, right? And so this is going to be called glaucoma, and that's increased intraocular pressure. So there's too much pressure inside the eye because we're making too much of that fluid. And I'm not going to ask you about the different types of glaucoma, but make sure you understand that a lot of times it has to do with a mismatch of production of that aqueous humor versus the reabsorption of it. When it does increase the pressure inside your eye, it can compress the choroid layer. Remember, the choroid layer is the back of the eye that has all the blood vessels that nourish the retina. And if we squish the blood vessels and close them, we can't get blood through, and so blood is not going to be able to go to and nourish the retina. And so this can cause retinal problems, reduced field of vision, dim vision, or halos around lights. And so that glaucoma can be an emergency. It's really painful too, so you'll probably feel it at least when it gets pretty severe. And this is one of those like eye emergencies that you want to try to get the pressure down pretty quickly. Now, as we said, the lens is this important little focusing area. Think about the lens in these glasses, right? So the lens in the glasses, they help change the way the light is refracted so that it's going to focus on the right part of your eye. So the lens is going to change the shape to focus light on the retina. So the cells in the lens are clear, they don't have organelles anymore, and they are just kind of full of this crystalline protein, and they're going to let light through, and hopefully our lens, as long as it's healthy, should be pretty clear so the light can go through it. As we said, the lens is connected to the ciliary muscles by these suspensory ligaments, and so it kind of holds it suspended in the middle of the eye, and if the suspensory ligaments are loose, it can kind of round up, and if they're tight, it's going to like pull it straight, and it's going to flatten the lens out. And the shape of the lens determines the degree of light refraction, so how much the light bends as it passes through the lens. And this is going to, once again, be determined by the ciliary muscle and the suspensory ligaments. And we can see here, this is going to be our lens here. The ciliary muscles are found here within the ciliary body. And then these are the suspensory ligaments. So when these muscles, they're muscles like this, they're sort of round. So when the muscles relax, right, they kind of relax like this, it's going to release tension on the suspensory ligaments, and that's going to stop pulling on the lens, and it's going to allow the lens to sort of round up. Whereas when the suspensory muscles tense, it kind of pulls them away. It's going to pull on those. I think I told you that backward, because when the muscle relaxes, it's going to relax, right? So this whole thing is going to relax. That's going to cause dilation. And I got that backwards, so I'm sorry about that. So listen to me now. When the muscles relax, it causes dilation, right? So it's relaxing. So think about like a sphincter. So when the sphincter constricts, it comes together. When it relaxes, it dilates. So when the muscle relaxes and it dilates, it's going to open up and dilate that ciliary process area, which is going to pull on those suspensory ligaments, and it's going to flatten out that lens. And this happens when you're viewing objects that are far away, versus when it gets constricted. It's going to constrict like a sphincter, and so that's going to actually cause this the diameter, the radius of this to get smaller, and so it's going to loosen up on the suspensory ligaments, and it's going to allow the lens to round up. I always get that confused because I think like, oh, when it's constricting, it should be pulling on the suspensory ligaments, but it's actually the opposite of that, because when it constricts, it's like a sphincter, and it actually constricts down and causes the lumen to get smaller, and therefore it's going to loosen up on those lenses and makes the lens more spherical. This happens when you view objects that are closer than 20 feet, and it's called accommodation. So in our lens, if we ever have opacities or like not clear areas inside our lens, we call those cataracts, and usually they can happen as a result of aging, but you can have congenital cataracts too that you're born with. And it causes reduced visual clarity, reduced color intensity, and they can remove these with lots of different techniques. So we've been using the word refraction a little bit. So refraction of light, what exactly does that mean? And this is going to be, it means how the light is bent as it passes like from one medium to the next. So as it passes like from air to water, as we can see here, or if it, as it passes through the air and the cornea, and also as it passes through a curved surface like the lens. And so the refraction is going to result from as the light passes from the air, it's going to go from air into your cornea, and your cornea is also curved, and then it's going to go through the aqueous humor, and then it's going to pass through the curved lens. And all of this is going to cause those light rays to bend a little bit. The refractive index, we don't talk too much about it, but is a number that represents the comparative density of different things. So this is just how, when we say the light is being refracted, we mean kind of how the light is bending as it's passing through different materials of different shapes and different densities. So as we are focusing our eyes, we can focus, it's really cool, your eyes are neat, you can focus on things far away, or you can focus on things close, and you can see, like, if you do that and you move your hands and you try to focus, you can tell that you're kind of moving your eyes a little bit, and you're using your eyes differently. So some things that happen have to do with changing the lens, but also with how your eyes are focusing. So we have something called the near response. So if we're looking at something close up, like your watch or your computer screen, anything within closer than 20 feet, the eyes do what we call converging. And so your medial muscles are going to be activated and the eyes are going to kind of look closer. So if you are looking towards something in your wrist, you can see your eyes kind of come together and you kind of look towards your nose. So your eyes converge. The lens accommodates. And so the ciliary muscle contraction is going to then contract like this, right? So it's going to narrow that area. It's going to slacken. It's going to loosen up the suspensory ligaments and allow the lens to thicken or round up. And then we're also going to constrict the pupil. And so when we constrict the pupil, as I said, it's going to make the pupil smaller. And that's going to limit the amount of light that comes through the eye, but also limit where the light goes. So the light is only going to be allowed to go through the center of the lens, which is like the most clear part of the lens. And so it's going to avoid the blurriness that comes from the edges. And so when we're looking at something close, our eyes converge, the lens accommodates, it rounds up because the suspensory ligaments are loose, and the pupil constricts. Now the opposite of this is the far response. So when we're looking at things far away, which I don't do enough, and that's probably one of the reasons that my eyes are getting so nearsighted, your eyes face forward, so they're not converged. You're looking straight ahead. So if you're looking far, like in a mountain far away, your eyes are not converged. You're not looking towards your nose anymore. The lens gets flattened, right? So the ciliary muscle relaxes, and therefore it opens up, and that's going to stretch on those suspensory ligaments and flatten out that lens. And the pupil is relatively dilated to allow in a greater amount of light into the eye. And so we can see here, this is going to be if we're looking at mountains far away. The lens is thinner, the pupil's a little bit wider, and then we're looking at things far away. If we're looking at something really close, our eyes are going to be accommodated, or excuse me, our eyes are going to be converged. The lens is going to be accommodated where it's thickened up like this. The pupil is a little bit narrower, so it only goes to the middle part. And then, once again, they should both focus right here on the fovea. And we actually focus things upside down on the back of our eye, and then our brain like knows that and can like flip them right side up, so we don't actually see them upside down. So we have some different what's called functional visual impairments. Obviously, I'm wearing glasses. I have some functional visual impairments. It's really common now, especially myopia or nearsightedness. So normal vision is normal, myopia. Hyperopia is actually also known as farsighted. This is when people have trouble seeing up close, but they can see really well far away. And that means your eyeball is too short. And so we can see a picture, maybe we can't see a picture. So your eyeball is too short. And so that would mean that when you're trying to focus on, instead of focusing right here, it's actually focusing a little bit back here because the eyeball is too short. Whereas we have, your book has a picture of it. I don't know if it made it into here. Myopia, this is like really common, and I think the incidence of myopia is increasing now as we go, in children too, especially, is trouble seeing far away objects because the eyeball is too long. So in this case, the eyeball would be too long. So the eyeball is out here, and then the like mountains that we're looking at would actually, are focusing somewhere in the middle of the vitreous humor. So if they're focusing in the middle of the vitreous humor, they're not really going to be in focus in the back of the eye. And we call that myopia or nearsightedness. So hyperopia can be corrected with a convex lens in glasses or contact lenses, whereas myopia is corrected with concave lenses like these. I'm very nearsighted, and there are a couple other things. Astigmatism, which is an unequal focusing, and that just has a little bit to do with unequal curvatures in like maybe your lens or your cornea and one or more refractive surfaces. And then presbyopia is the age-related change in vision. That's going to be really common. Most people have this happening as they get, into their later ages. Your lens is not able to round up and become as spherical as it was before, and so it can't accommodate well. Remember, we needed accommodation to see things up close, and so reading close upwards can become difficult, and you can correct that with convex lenses. Now, if you already had you know, myopia, you need concave lenses for that, and then you get presbyopia, then you might need bifocals as you go. But all of these things can be treated with various surgical techniques. Now they have LASIKs and different things like that. All right, so here's the picture I was talking about earlier when I was mentioning the different layers of the retina. So now we said that we are taking all of that light, it's passing through all those structures, we're refracting it, and we're trying to hopefully focus it directly on the fovea centralis in the back of the retina. So we don't want it to focus in the middle of the vitreous humor. We don't want it to focus like somewhere out behind the eyeball. We want it to focus directly on the fovea centralis right here. So that's the goal, and sometimes you might need corrective lenses in order to cause that to happen. So when it does, we're going to have light focusing on the back of the eye, and then this is the neural part of the retina. So now we're going to kind of talk about what happens in the retina. So first we have this pigmented layer here. This is going to prevent light scatter. It's going to, by absorbing some of that light here. These are the photoreceptor cells. The rods are yellow, the cones are blue. The next layer is the bipolar cells. And then finally we have the ganglion cells. And we'll talk about these cells as we go through phototransduction. Other parts of the retina. So we said this is the fovea centralis here. This is where we're trying to focus everything, especially when you're reading or seeing anything that's really in color and nice sharp relief. The macula is the whole area, and the fovea centralis is the very middle part. And this is the area that if people have macular degeneration, this is the area that starts to, those cells in here don't have a good blood supply and they start to degenerate. We also have this area right here where all the blood vessels go. They're going to what's called the optic disc. And this is the back of the eye where all of the axons of the ganglion cells are exiting and where the optic nerve and the blood vessels are exiting from. Because of that, like everything is exiting there, there aren't actually any rods and cones there. It's just a bunch of axons exiting. And so we can call this the blind spot also. You actually can't really, this doesn't have photoreceptors in it, so we can't see in this part of the eye. The peripheral retina, so everything on the outside, is going to be mostly rods. So this is going to be mostly rods, and then we have some rods in the fovea too, but we have most of our cones here. So the periphery or the outside part of the retina is going to have mostly rods. So the retina is the very back of the eye, and it actually sits on top of the choroid, but it's not really as connected as like it could be. The vitreous humor is going to be pushing it back and squishing it onto the choroid. But sometimes the retina can become what's called detached, and so it kind of floats away, or maybe some of the vitreous or some stuff gets in between the retina and the choroid. The outer pigmented and the inner neural layers are going to kind of separate so that the neural layer with all the photoreceptors kind of comes away. And these neural layers are now far away from the pigmented layer, and they're far away from the choroid with all the blood, and so they become deprived of nutrients, and that's not good because those neurons need nutrients. And so it can look like there's a curtain coming and like covering your eye. You can also have an increase in what's called floaters, and I have floaters all the time, unfortunately. But they're little things that, like, if you look at bright light, you can see things floating in your eye. A lot of times they have to do with like little pieces of the vitreous humor that are kind of clumped up, and light is kind of passing through them. If you are really nearsighted, you'll usually have more of them, or obviously if you're getting detached retina. Generally, if you're nearsighted, your eyes are getting longer and longer, or they are like longer and longer. And so the more nearsighted you are, like once you get into like the plus, 5 or whatnot, probably as nearsighted as I am or more, you do have an increased risk of retinal detachment. But if you do have a retinal detachment, then they do have treatments where they can actually like stick the retina back to the back of the eye. So they do have to do it quickly though. So this is another one of those emergencies. All right, now we're going to move to phototransduction. This is kind of the important part where we're saying, okay, now the light has gotten in the eye and we focused it on the back of the retina and we talked about all those structures. How exactly do we turn this light into an electrical signal? And that is going to be performed by those photoreceptors, so the rods and cones. So that's kind of like that innermost layer of that neural retina. And so if we look at the part of a photoreceptor, they've got a couple of different parts. So one of them is the outer segment. And so here we can see this area here is the outer segment. Then we've got the inner segment and the cell body, and these are the synaptic terminals. So kind of remember this. I'm going to be flipping back and forth between this. This one is a cone because it's cone-shaped. This one is a rod. It's rod-shaped. You can see that the outer segments are embedded in the pigmented layer of the retina. And the outer segment is going to have lots of photopigment containing discs that absorb light. And we'll talk about what photopigment is, and we'll look at the discs in a little more detail in a second. The discs are going to, these are kind of replaced often. So we'll have the discs degenerate and then we'll get new ones made. And we get, the photopigment gets replaced too. The inner segment contains the cell's organelles, right? The mitochondria, all those important things. The cell body contains the nucleus, and then the synaptic terminals contain those vesicles storing the neurotransmitter. And this neurotransmitter is called glutamate. So we haven't really talked about glutamate that much yet, but the glutamate is the neurotransmitter that the bipolar cells, or excuse me, the photoreceptor cells use. So we said that there are two different types. We have rods and cones. Remember that cones are good for color visions, right? They both start with C. So cones are also concentrated at the fovea centralis, and they allow for color vision. But in order to have good color vision, you need a lot of light. So cones are only activated in high intensity of light. So if you've ever walked around your bedroom at night, everything looks blurry and everything looks black and white, right? You can't see color very well if you don't have enough light because your cones need light to be activated. In that really dim amount of light, like when you're walking around your bedroom, you're really only using the rods. Rods are very sensitive to light, so they're activated in dim light, but they don't see color. You kind of see the black and white. They're also not as clear, so that's why it looks really blurry when it's dark, because they are not, they make a blurry image. So many rods converge on a few bipolar cells. So you have a lot of rods that go to just a couple bipolar cells, and they are going to converge on just a few ganglion cells. And so each rod, it's not as precise in its localization. It's going to give more of a blurry image. So it's very sensitive, but gives you a blurry image that does not, and you don't see color. And we've probably all experienced that. Whereas cones have a one-to-one relationship with bipolar cells and ganglion cells. So each of them have their own bipolar cells and ganglion cells. So they're very they give you a very sharp image, but since they're only activated in high light intensity, it's only possible in bright light. And that's same with colors. You can only see the colors with bright light. And most of those are focused at the fovea centralis. So here are the rods. They're the blue one, or excuse me, the cones are the blue one. They're cone-shaped. The rods here are going to be more rod-shaped. They're going to contain different types of pigments in their little discs. So here are the discs. These flat discs are going to be made of normal cell membrane. And within the cell membrane, we have this photopigment. And this photopigment is embedded in these discs. And this is where the actual light is going to be causing a change. Because we're like, how does light open up a channel or do something to make, to depolarize the cell? Light can't stretch the cell. Light doesn't, there's not a chemical to activate a chemical receptor. Light doesn't, we're trying to figure out what light exactly does to activate the cell. And it actually has to do with this pigment here. So this photopigment is going to be reactive to light. And this is how light is going to cause a change in these receptors. And so we're going to look at this photopigment up closer. A photopigment is a light-absorbing molecule. And so it absorbs the light, and when it absorbs the light, it's going to change shape. These are found in the outer segments of both rods and cones, but we do have different photopigments in rods as we do in cones. They're made of an opsin protein and a light-absorbing retinol. And this retinol is made from vitamin A. So it is important that we have enough vitamin A. That's why we say vitamin A is important for vision. It's important for these photopigments. Different pigment types have different opsins, and they transduce different wavelengths or colors of light. And so each pigment is going to react to different colors of light. And so rods contain a pigment called rhodopsin. And then we have three different colors or types of cones. So we have blue cones that detect short wavelengths, green cones that detect and absorb sort of intermediate wavelengths, and red cones that detect long wavelengths. And we can see the rhodopsin one is kind of in intermediate one. It's kind of in the middle. But then we have blue cones, green cones, and red cones. And red-green are closer together. And then you can kind of remember that because you can be red-green colorblind, so they're kind of closer together on this wavelength scale. So we have, the cones have 3 different types of opsins, red, green, and blue, whereas the rod just has that one rod opsin. And when we talk about phototransduction, we're going to use rods as like the example, but know that the same thing happens in cones, just at different wavelengths. And we said color blindness, just like I mentioned the red-green color blindness earlier, is an X-linked recessive condition. And so since it's X-linked and recessive, it's more common in males. And it's usually the absence or deficit in one type of cone cell. And so usually it's red or green that's most commonly affected. And so it makes it so that you have difficulty distinguishing red and green. So you just got the 2 different ones instead of 3. So when we look at our photopigment back here, so I just want to go back to here. Retinol is this little guy in here, and then opsin is going to be this kind of pigment here. And remember that we're going to have different opsins for different wavelengths. So rods have one called rhodopsin, and then the cones have the photopsins, the different colors of photopsins. So we're going to use rods and rhodopsins as our example here. And so phototransduction is how light is converted to the electrical signal. And remember, we need to convert this light to an electrical signal because the brain uses action potentials or electrical signals. It can really only read electrical signals. And so in order to do this, light has to cause a change in the cell that eventually causes an action potential or stops an action potential, depending. It's going to change something in the cell. So in this case, phototransduction is like backward sometimes. You'll see what I mean as we go through. But remember that we're going to say in the dark, we're looking at rods as our example. In the dark, rhodopsin contains what's called a cisretinol. And light is going to cause this cisretinol to change into a transretinol. And these terms just have to do with the folding, how this retinol is kind of folded. And so it goes from cis to trans, and it's going to change. And so it changes its shape. So know that it changes its shape. When it changes its shape, it kind of disassociates from the opsin. And so it kind of kicks off the opsin. They split up, right? They changed its shapes. They don't fit anymore, and they split up. And this is called bleaching. So because it was a pigment, and we're breaking up the pigment, so bleaching would be like we're getting rid of the color. It's called a bleaching reaction. So we can see this right here. So here's our, in the dark, we have our rhodopsin is all together. We have the cisretinol that has this little bend in it. When the light comes in at the correct wavelength of light for rhodopsin, then our retinol is going to absorb the light rays, and it's going to straighten out that kink, and it's going to change the shape of, and it's going to change the cisretinol into transretinol. And then transretinol has changed its shape, and it doesn't really fit anymore with the opsin, and so they disassociate. And that means they're going to break apart. And we call that the bleaching reaction. And so this is when our cis and transretinol break apart. That's the bleaching reaction. And so that happens in the light. So these pigments are bleached and broken apart by the light. After bleaching, the rhodopsin must be rebuilt for the rod to function again. And so the transretinol is transported to the pigmented layer, and then in the pigmented layer, we convert it back, we bend it again to the cisretinol, and then it's going to be transported back into the rod and combined with the opsin to make that photopigment again. The process is really slow for rhodopsin, and so the rods generally don't function in bright light because the bright light is constantly bleaching them, and so they're always bleached, and they don't function very well in bright light because there's always, they're always trying to rebuild those photopigments. Process is similar for the cone photopsins, but quicker. And so we need a more intense light to bleach your eyes, and they regenerate fairly rapidly. And so here, once this has happened, then the transretinol is converted back to cisretinol, and then it's reconnected to the opsin, and then we have our pigment back together again, and it's ready to be bleached again. And this has to do with adaptation, light and dark adaptation. So when you're in the bright light, your rods are being bleached, right? Your rods are constantly being bleached. You don't have enough rhodopsin rebuilt to really have them regenerated yet. And so all like in bright light, because remember they're really sensitive to light, pretty much all your rods are going to be bleached. And so when you then turn the light off in the dark, your rods are all bleached, so they're not working very well because they still have to take a little while to regenerate. And your cones are not sensitive enough. Like your cones don't work in the dark. And so you're kind of pretty, like, right when you go from bright light to dark light, you're pretty blind for a while because your rods are still bleached from being in that bright light for a while, so they're not working. And your cones don't work in the dark. And so because it takes a little while to regenerate that rhodopsin, it takes a while before your eyes adapt to the dark. You know, if you've ever gone in, like, I don't know, if anyone's ever used real old x-rays where you had to go into the room with the red light and run them through the x-ray developing machine, it takes a while for your eyes to adjust into the dark. The opposite of this is light adaptation, the process of adjusting from low light to bright light conditions. The pupils constrict, but the cones are initially overstimulated. So when we have really bright light, if you ever go outside, your pupil is wide at first, and you go outside and it's really bright, and so it lets in way too much light. And initially your cones are overstimulated, and some of them might get a little bit bleached out, like you have that really bright light that you see in front of your eyes. And it can take a little while to fully adjust. All right, so we kind of talked about that bleaching reaction and what that does, but now how does it actually cause an action potential? So we're going to move on to that next. So remember these, the opsins. So this is going to be our photopsin, in photopsin in cones and rhodopsin in rods, and we're going to use rods as our example. And they're going to be activated by light. And so we're going to talk about what happens in the dark. So when it's dark, we are going to have our rods with the rhodopsin are normally depolarized. So the membrane's at negative 40 millivolts, which is, you know, less than threshold. So in the dark, this is backward. This is why I said I feel like phototransduction is backward for me. Rods are depolarized. And something called cyclic GMP, or a cyclic guanine monophosphate, is produced. And cyclic GMP binds to cation channels and keeps them open. These cation channels, remember, are going to let in cations. And in this case, we're going to have sodium and calcium are both entering the cell. And this is called the dark current. So in the dark, this is what's happening. Voltage-gated calcium channels in the rod's synaptic terminal stay open, and glutamate transmitter is continuously released by the rod. Now, glutamate is an inhibitory neurotransmitter. It's going to cause an IPSP in the bipolar cell, and it prevents them from exciting the ganglion cells. And so we can see this picture right here. This is what happens in the dark. Normally, we have this cation channel is open because we have high levels of continuously produced cGMP. So just like that's the baseline. We're making this cGMP. It's keeping the cation channel open. It's letting in positive ions. Those positive ions are depolarizing the cell and causing action potentials, right? These action potentials are going to cause the opening of the voltage-gated calcium channels, and that's going to cause the exocytosis to happen, and glutamate is going to be released. Now, so in the dark, we're always releasing this glutamate from our rods and cones. And this glutamate right here is inhibiting the bipolar cell. And because it's inhibiting the bipolar cell, it's not talking about it's not going to be activating the ganglion cell, and there are no nerve signals generated. So we're saying this is really dark. The rods are not activated. We have no nerve signals generated. So when it's really dark and there's no activation happening, then the rods inhibit the bipolar cell and there's no signals to the brain. So the brain says, nope, there's no light, it's dark. Now what happens in the light is kind of weird. It's the opposite. So in the light, then the light is going to cause hyperpolarization of the rods. And how does this work? So light splits the rhodopsin. And when that happens, so this splitting part here, when the light splits this rhodopsin right into this transretinol and this opsin here, we said that that's going to be called that bleaching reaction. That bleaching reaction is going to activate a second messenger system. And this second messenger system is going to cause something to happen in the cell. It's going to activate phosphodiesterase, and it's going to break down the cyclic GMP. And what was the cyclic GMP doing before? The cyclic GMP was actually keeping open those cation channels. Now that we have less cyclic GMP, the cation channels are going to close. And they're going to prevent those positive ions from entering the cell. And so once those positive ions stop entering the cell will eventually become more and more negative. And as it becomes more negative, it's, that's like an IPS. It's not depolarizing anymore. It's not going to reach threshold. We're not going to have an action potential, and we're not going to have, and so those voltage-gated calcium channels in the rod synaptic terminals are going to close. So the rod stops releasing glutamate. But remember that glutamate was inhibitory. Glutamate was the thing that was stopping the bipolar cells from having an action potential. So the bipolar cells now are like, oh wait, there's no more IPSPs. And they're going to create their own, they are then going to depolarize on their own. When they're not inhibited, they're going to depolarize. And so when they're not inhibited, they're going to depolarize. They're going to release their own neurotransmitter to the ganglion cell. And that's going to be excitatory. The ganglion cell then is going to be excited, and it's going to lead to the impulse being sent along the axon to the brain. And so here, because the bleaching reaction has happened, it caused a second messenger system that causes that phosphodiesterase to decrease the cyclic GMP. So we don't have cyclic GMP to bind to and open these channels anymore. So when the rods get bleached, the downstream eventual effect is that those cation channels are going to close. That's going to stop depolarizing the cell. The dark current goes away. It's not positive anymore. There's no more action potential. We are not releasing glutamate. And remember, glutamate was inhibitory here. It was inhibiting the bipolar cell. So once we stop releasing this glutamate, this bipolar cell is like, yes, I can start depolarizing now. It's not inhibited anymore. And it's lost its inhibition. So it's going to excite the ganglion cell, and the ganglion cell is then going to send information down to the brain. And so it's going to say, okay, now you see things. That's the light. There is light. So it's sending signals to the brain. So in the dark, the photoreceptors are depolarized, and they are actively inhibiting nerve signals to the brain. In the light, the photoreceptors are hyperpolarized, and they stop inhibiting the bipolar cell, and so we do have signals to the brain. As I said, I feel like it's a little bit backward. So normally you would think that, the light would cause an action potential in the rod, but it's actually opposite. So just remember that. So in the dark, the rods are depolarized, and the action potentials are happening in the rod, but they're inhibiting the bipolar cell. And therefore there's no nerve signals. Whereas in the light, the rods are hyperpolarized. There is no inhibitory glutamate, which lets the bipolar cell excite the ganglion cell. And that's going to send information through the axons of that ganglion cell. And remember, all those ganglion cell axons are going to go out, and they're exiting the eye as the optic nerve, right? Cranial nerve 2, because you have 2 eyes. Sorry, that's... We're going to go straight to visual pathways now. So in the retina, first the light is going to hit the photoreceptors, right? And then that signal is going to go from the photoreceptors. It's going to stop inhibiting the bipolar cells. The bipolar cells are then going to excite the ganglion cells. And that ganglion cells, those axons are going to exit at the optic nerve. And so the optic nerves, you have 2 of them, 1 for each eye. They come together and they converge at what's called that optic chiasm, right? That sits at the very base of the brain. And so let's see if this picture right here, here's the optic chiasm. So you can see the right eye here is going to be these blue fibers, whereas the left eye here has these red fibers. So at the optic chiasm, the medial fibers are going to cross. And so the medial fibers from the right eye are going to cross and go to the left side of the brain, whereas the medial fibers from the left eye are going to cross and go to the right side of the brain. But the lateral fibers stay ipsilateral. So the lateral fibers from the left eye go to the left side of the brain. the lateral fibers from the right eye go to the right side of the brain. So on the optic nerve, we have all the fibers from each eye, right? So we have the right optic nerve and the left optic nerve. At the optic chiasm, some of those fibers cross. Then they're going to go back to the optic tracts. The optic tracts are then going to be mixed. So they contain axons from some axons from the right eye and some axons from the left eye. So each optic tract is going to have both of the information from both of those eyes. So as the optic tracts, they are now in the central nervous system. They kind of go back around the brain. Most axons are going to go to the lateral geniculate nucleus of the thalamus. And then that's going to go to the visual cortex in the occipital lobe. Remember, the thalamus is that sensory sorting area, so lots of things go there first. Some of them project to the midbrain, though. Some of them go to the superior colliculi. Remember, the superior colliculi are the top of that textile plate that we identified in lab, and they are going to coordinate reflexive eye movement. So like if you see something, your eye can reflexively move. There's also another nuclei called the pretectal nuclei that is going to be important in the pupillary light reflex and the lens accommodation reflex. So both of those are like those internal reflexes inside the eyes. The other thing is that the ganglion cells are also, they have their own pigments. They're photoresponsive and they have their own pigments. And these are going to also they send other information to the pretectal nuclei. And remember that the amount of light that you see, and like the amount of blue light especially, can affect your circadian rhythms too. So that's going to have some effect on there too. So you have an overlapping field of vision. And so your right eye sees only some part of here, your left eye sees only some part of here, but in the middle, you have an overlapping binocular vision. This is going to help us with depth perception. So our visual fields are going to go back from the eye to the optic nerve. They're going to cross the optic chiasm. Some of those axons are going to cross and some won't. Then it's going to go back to the optic tract. Some of it's going to go to the thalamus and then back to the visual cortex for like direct processing so we can figure out what we're seeing. But then some of them are going to go to the reflex centers in the midbrain, like the superior colliculi and that pretectal nucleus for reflexive eye movements, and then, you know, the pupillary light reflex and accommodation reflex. So visual pathways, as I said, can be kind of complicated. I think that the most confusing is this part right here, the phototransduction. I always get this backward because I feel like it's counterintuitive. You'd think that the light would cause the photoreceptor to depolarize, but it's not. It's already depolarized in the dark, and the light actually stops it from releasing an inhibitory neurotransmitter. It's just kind of a weird thing, but it's interesting, and it's cool to figure out. This part is a little bit confusing too, and how that associates with phototransduction. So I think that make sure you know the structures. Watch the anatomy lab. Watch this one again if you need to. Phototransduction is probably the most kind of like complicated physiology one that we'll talk about. And then make sure that you know the path that light takes through the eye, as well as the visual pathways here.